Okay, so 2024 AP Biology free response questions. This is readily available on the web, on the AP website. I hope that you guys download this file and solve it on your own before actually watching this video. It will give you a better sense of how you're doing in the course. Of course, it depends on when you're watching this video. If you're watching this video in April or early May, then yeah, I think that's very useful. If you're not, if you're watching it in January or February where you're starting to prepare, then yeah, maybe, being able to solve this entire set might be a stretch because you haven't finished the whole course yet. But regardless, come join me for about half an hour. Hopefully, we'll be able to get through this test during that time. And in the interest of saving time, I'm going to skip over the equations and formulas. I don't think you really need to use these too often. If you do, it'll be very specific. It wouldn't be the entire thing. So we're going to skip over that and get straight to the questions. Now, this 2024 is really the first year that they've reformatted the exam such that they give you a little bit of information and then they ask you about it. And then they give you a little bit more information and they ask you about that. Before, all of the information was just front-loaded and then all the questions came later. I'm not really sure how much I like this. Maybe I'm just not used to it, but it's okay. I think it really makes sense for this first part of the questions, and we'll dive into that right now. So number 1 says, crossing over in meiosis 1 is required for homologous chromosomes to properly align during metaphase and segregate during the first division. What I'm going to try to do for this video is to put up some text overlay on the video to kind of let you guys know what unit from AP Bio this meiosis is coming from. By the way, this is Unit 5 on heredity or inheritance, and also link it to some of the chapters in the Campbell biology textbook. Now, I know depending on the edition, it might change, but this is typically, I think, like chapter, I want to say 13, meiosis. I think it's chapter 13, meiosis and the sexual life cycle. So I'll try to do that. So hopefully it's on the screen right now. Anyway, so crossing over in meiosis 1. Okay, so that's kind of what we're talking about. But the question itself, the first one, 1A, asks something a little bit different. It says, describe the function of S phase of interphase. Now, in one of my earlier short-form videos, I actually talked about the fact that even for meiotic events, it's preceded by the interphase. And the duplication of DNA that occurs during the synthesis phase of interphase is necessary for even what we're going to be talking about here, crossing over to occur. You need to have those sister chromatids that you have here at the top and sister chromatids here that you have at the bottom in order for you to do that crossover exchange. So this S phase is going to be very important. And when you have to describe the function, you just simply have to say that that the synthesis phase is responsible for the duplication of DNA prior to cell division, and that should be good enough. You don't have to necessarily link it to meiosis in this particular case. But moving right along, it says, some regions of a chromosome called hotspots display a higher frequency of crossing over than other regions do. Crossing over is suppressed in chromosomal regions near the centromeres. So centromeres are these regions at where the two arms are being held together. The centromere region of a duplicated chromosome includes a collection of proteins that form a structure called a kinetochore. Great, so we know that kinetochore is where those microtubules or spindle fibers attach to. So we've seen that term before in, I would say, just even in cell division, mitosis in chapter 12. Scientists hypothesize that one or more of these kinetochore proteins are responsible for suppressing crossing over around the centromere. So here we're given some new information that is not typically found in the textbook or even taught by most teachers, but they just tell you here that near where the kinetochore proteins are, you actually have fewer crossover events. And I guess if you look at what they say about hotspots, here we actually have more crossover events. So this actually goes to show that crossovers, while they are random, can be suppressed in certain regions for various reasons, I suppose. And they're not going to tell us the reasons why near the centromeres crossing over is suppressed, but we just have to take it with take it as is and just go with the flow. Because they're going to actually tell us a little bit more. They say to investigate their hypothesis, scientists modify chromosome 8 in yeast such that in each cell, one chromosome from the pair of homologous chromosomes 8s contained gene encoding red fluorescent protein, RFP, while the other chromosome from the pair contained the gene containing green fluorescent protein, or GFP. Hopefully you've seen GFP before. I think we see this in transformation experiments often. Cells expressing RFP emit red light, and cells expressing GFP emit green light, naturally. Models of the modified chromosome 8, both before and after crossing over, are shown in Figure 1. So let's quickly take a look at Figure 1, and it shows here before crossing over in A, and you can see that one of the homologous chromosomes has the RFP, the other has the GFP, and incidentally, they put these genes on either side of a hotspot, so that's kind of interesting. And after crossing over, yes, so we see that there was a crossover event between this chromatid and this chromatid, such that the gene encoding for RFP has now moved over to the bottom chromosome, and now it's found on this chromatid on either side of the hotspot here. So we have the GFP and RFP. That's basically what crossover is anyway, right? Okay, so the second part says explain why some haploid cells formed from meiosis in this experiment will have just one fluorescent marker. So this is talking about the haploid cells which ultimately result from this meiotic event. So as you guys know, if we're just looking at this particular diagram right now, this top homolog is going to go up to one of the cells after meiosis 1, and the other homolog will go to the other cell during meiosis 1. But in meiosis 2, each of these homologous chromosomes will be in separate cells, and the sister chromatids will actually separate during anaphase, resulting in each of the haploid cells containing one of these chromatids. So even if we just extend that analogy, like visually, let's say, we could already see that if what we have here in this example image is what we go into meiosis 1 with, or, well, let's say anaphase 1 with, then the daughter cell, or the gamete, that includes this chromatid at the top, will only have that one RFP encoding gene. The bottom one will have no fluorescent protein. The top one from the bottom chromosome will have 2, and then the bottom one will have 1. So that totally explains why some will only have one fluorescent marker. And one of the ways that we can say that is, given that crossing over can result in parental combination or parental chromatids that do not undergo a particular crossover event, or alternatively, an even number of crossover events. Because if you think about what an even number of crossover would do between this GFP and the RFP, it will sort of shift it once, but then shift it back, resulting in, well, I mean, I'd have to sort of use this top example, but it was shifted and then shifted back, resulting in, once again, just having the original GFP or the RFP gene in the chromatids without doubling up or not having one at all. So I think that is going to be the explanation for this answer. So that would be either parental combination of genes along the chromatid or an even number of crossover events, resulting in the return to the original configuration. All right, let's move on to the next part. It says the scientists then investigated whether attaching individual kinetochore proteins to a specific DNA sequence present in a known crossing over hotspot on chromosome 8 affected the frequency of crossing over at this location. In their first experiment, they examined 3 groups of yeast cells containing the modified chromosome 8. Group 1 contained no kinetochore proteins attached to the hotspot. Group 2 contained kinetochore protein CTF attached to the hotspot. And group 3 contained the kinetochore protein IML attached to the hotspot. And for each group, the scientists determined the frequency of crossing over between RFP and GFP genes. To determine the frequency, the scientists added a number of cells emitting both red and green light to the number of cells that emitted no light and divided the total number of cells. Now, this last part doesn't really matter too much to me. For me, they actually just tell you on the y-axis the frequency of crossing over. So that's really good enough, because here in the graph we see that the cells, or let's say chromosomes, that didn't receive any artificial kinetochore proteins in the hotspot had a frequency of crossing over around 7.2%, whereas the attachment of CTF onto that area resulted in a reduction in crossing over. And we have IML, which doesn't seem to be affected even with the attachment of those kinetochore proteins. Now, what's really important to note in graphs like this is that the averages are not really the important bits. What's really important is what we call the spread of the data. And the spread of the data, or the central tendency, is typically illustrated by these error bars. And these are typically two standard errors. And normally what we do in a test like this, so this would be usually like a analysis of variance or an ANOVA test. If you're taking AP Stats, you know what that is. As long as the error bars do not overlap, we can say that they are actually statistically different. But in this case, I think we would argue that none, so no kinetochore proteins attached to the hotspots, and IML are not statistically different. So it almost means that IML has like no effect on the amount of crossover event that would occur even if they were attached to the hotspot, whereas it's pretty clear that CTF has a significant reduction in the number of crossing over or the frequency of crossing over that occurs there. So let's actually take a look at what kind of experimental questions they're going to ask with B1. It says, identify the control group for the scientist's first experiment shown in figure 2. So here we have figure 2, this none, right? Or at least the chromosome 8 that did not receive any kinetochore proteins, that would be our control. In a follow-up experiment, the scientists created a modified version of CTF in which the DNA binding portion had been removed. They compare the frequency of crossing over in yeast cells in the presence and in the absence of unmodified CTF with that in yeast cells in the presence and absence of the modified CTF, and the data is not shown. In the follow-up experiment, justify why the scientists use a modified CTF protein that is unable to bind to DNA as a control. So we know from looking at figure 2 that CTF absolutely decreases crossing over given that it's attached to the hotspot region of the DNA, but we don't really know how they did that. For example, whether they just simply mixed the CTF with the solution containing DNA, I think that's what they're getting at here, right? So for example, is it just CTF present in that solution that would cause a reduction in crossing over, or does that CTF have to bind to the DNA and actually operate at the level of DNA, in conjunction with DNA rather, in order to have an effect? And I think that is exactly why they want to use it. They want to see the interaction between CTF and DNA rather than simply the presence of CTF and DNA. And that is going to be the best answer or the best justification of that question. The last one is identify the independent variable in the follow-up experiment. Of course, the independent variable is what we choose. So this would be either it would be the use of a modified CTF or the use of an unmodified CTF. That would be our independent variable. Moving on to C. Based on figure 2, describe the effect on the frequency of crossing over when CTF is attached to chromosome 8 compared to when IML is attached. So this is something I alluded to earlier. So in this scenario, we know that statistically speaking, it seems that CTF has an impact. It reduces the number of crossing over events, whereas IML does not actually significantly reduce. And of course, that difference is pretty clear from the non-overlapping error bars that we observe in the bar graph. D, predict the effects on the number of copies of chromosome 8 likely to be present in the resulting daughter cells when CTF is attached to the hotspot. Now, this is an interesting question to me because it seems that now they're asking whether the attachment of CTF in the hotspots will have an impact on the proper disjoining of sister chromatids during meiosis II. This is something that we call non-disjunction, as in if the two sister chromatids do not disjoin properly during anaphase, what you would see is a potential trisomy in the offspring that would be formed, I guess, from the gamete that would have the double chromosome, in this case, chromosome 8. The answer to this has to be given in the text. And the reason I say this is I don't think this is something that you necessarily learn in AP Bio. So I'm going to go back up here and read you guys one text. It says crossing over in meiosis I is required for homologous chromosomes to properly align and segregate. Okay, now why is this an important fact? Well, because if we go down here, we know that CTF reduces crossing over. So if crossing over is reduced, then perhaps the segregation of those sister chromatids could potentially be affected as well. So the best prediction here is that some of those daughter cells will have either double copies or 2 copies of chromosome 8 or no copies at all because, well, you know how non-disjunction could result in one gamete without a chromatid. So that would be the best answer here. I kind of provided the justification for that, so I'm just going to set that aside for now. Let's go to D, 3. Explain how the presence of hotspots could increase the likelihood that a population will survive in the presence of selective pressures. So wow, we jump straight over to Unit 7. This is from evolution, because what we know about sexual reproduction, as we introduce it in Unit 5, is that sexual reproduction sort of evolved in order to maximize the variation in the offspring that we produce. So one of the ways that we did that was through crossing over. So if hotspots increase crossing over, that would mean that what we're trying to do here is to maximize the amount of recombination for each of the meiotic division events, such that all of the gametes that we produce would be very distinct from one another. And in that case, selective pressures, which is typically associated with the environmental pressures and, of course, changing of that environment, would be mitigated by having as many variants as possible in the next generation, such that even if the selective pressure changes, or even if it remains static, by having numerous genetic combinations in your offspring, maybe one or maybe more would be likely to survive and carry on your genes to the next generation. And I love it when they do this. Evolution is so much fun. Okay, so that's number 1. Great. Let's move on to number 2. To investigate how increases in environmental temperatures affect the metabolism of certain organisms, researchers incubated liver cells from toads at different temperatures and measured 2 markers of metabolic activity: the rate of oxygen consumption and the rate of ATP synthesis. So before we dive in any further, I'll just add that this is from Cell Energetics, so this would be Unit 3, and Chapter 9. Chapter 9 is cell respiration. Now, it's funny because, the AP curriculum usually teaches photosynthesis first, but the chapter layout of Campbell is, cell respiration Chapter 9 and photosynthesis Chapter 10. But I'm digressing, so let's actually get right to it. Oxygen is the final electron acceptor in the electron transport chain. So consumption of oxygen is directly correlated with, ATP synthesis. So these are actually just kind of like the same thing. And we see that correspondence here. Temperature is going to be our independent variable. As we increase the temperature from 20 degrees, 25 degrees to 30 degrees, the toads are consuming more and more and more oxygen, and they're producing more and more and more ATP. Of course, they're in different units. Oh, actually, they're not in different units. Oh, that's interesting. Oh, that's really cool. Yeah, I would have never thought that these would have such high correspondence even in the same units, but hey, that's kind of cool. All right, that doesn't matter. Let's go back to A. Okay, describe the role of water in the hydrolysis of ATP. Well, it's in the name, isn't it? Hydro meaning water, lysis meaning cut. We know that in the chapter 5, where we learn about polymers, anytime you break a biological macromolecule, you use water in a process called hydrolysis. Now, I'm not saying adenosine triphosphate is a macromolecule. It certainly can be a component of it. But even when we take ATP and we break off that phosphate, we would still use water. So it's used to cleave the bonds between the phosphate and, of course, the resulting adenosine diphosphate. Now, moving to B, though. Using the template in the space provided for your response, construct a bar graph that represents the data shown in Table 1. Your graph should be appropriately plotted and labeled. I'm going to actually post a picture of this, probably linked in the description below, because I don't really want to draw this on an iPad and then link it to the video. I could, but it's going to take a lot more time for you guys to watch me do that. It's kind of boring. So I'll post it here. But what's important is that based on that graph, we will be able to answer the next question. I'll explain what I mean by this. Because it says, based on the data, determine the temperature in Celsius at which the rate of oxygen consumption, so this box over here, is different from the rate of oxygen consumption at 25 degrees. So if we use 25 degrees as our standard, which one of these temperatures, whether it's 20 or 30, is actually different? And I like how they say just different, because we know what they mean when we say different. In science, when we say different, we mean statistically different. And that means the error bars should not overlap. So here's one fun way to do it. Let's take 20 degrees, for example. We have 12.8. Now we have plus and minus 2.2. That's the size of the error bar. So you can imagine that if I were to go up from 12.8, 2.2, then I would have 15. Now, if I go to 25 degrees and I go to the below 16.5 with the error bar, then I'll get 14.5. Notice how that the upper end of this data and the lower end of this data point are going to overlap. So that's not different. Now, because 30 degrees, we know that the trend is that it's going to be higher. I'm going to go to the upper bounds of 25, and that looks to me like 18.5. And the lower bounds of 30, that's going to be something like 21.4. I think that's right, 21.4. Yeah, those do not overlap, meaning that yes, 30 degrees and 25 degrees are, in fact, different, not just in terms of the means, but in terms of statistical significance. They are really different indeed. Now, moving on to C, based on data in Table 1, describe the effect of temperature on the rate of ATP synthesis. Well, that is clearly that the warmer the temperature, the greater the ATP synthesis is going to be. And then we have to calculate the average amount of oxygen consumed in nanomoles for 10 milligrams of mitochondrial protein after 10 minutes at 25 degrees. So we're going here at 25 degrees, and here we have 16.5 nanomoles per minute per milligram. So what you would have to do is you'd have to first of all multiply 16.5 by 10 to get to 10 milligrams, and then multiply that by 10 again to get 10 minutes. So it's really 100, right? You're multiplying this by 100, which means that you'd have something like 1650. I think that sounds right. Nanomoles, okay, of oxygen consumption. And that's an absolute number because we're looking at it in particular time and particular temperature. Let's go to D. Oligomycin is a compound that can block the channel protein function of ATP synthase. Now, guys, you guys know where ATP synthase is. This is found in the inner membrane of the mitochondria. It's sort of at the end of the ETC. It's not technically part of the ETC, but as the electron transport chain builds that proton gradient from the electron's energy that's arriving from NADH and FADH2, it's going to be pumping those protons into the intermembrane space, and it usually falls back into the matrix, and it does so with ATP synthase. So we're going to block that. So no protons are falling back in, and I think it's presumed that ETC is still doing its thing. So it said predict the effects of using oligomycin on the proton gradient across the inner mitochondrial membrane. So, okay, if the ETC continues to do its thing, pumping protons into the intermembrane space, but it's not coming back down through ATP synthase into the matrix, then of course the proton gradient will become greater and greater and greater. And that could be that the intermembrane space could be more acidic. Like that would be another way to think about it. In any case, yeah, that's basically it. Now the next one is going to be justify your prediction. Well, so it's something that I just sort of explained as we addressed this question from the beginning, so I won't do that. You could just roll back the video a little bit and figure out what the justification is going to be. Great, so those are the two long-form questions, and they weren't that bad so far. Let's move on to the next one. To investigate whether red blood cells of animals lose the ability to take in glucose from their environment as they age, scientists collected red blood cells from guinea pigs that ranged in age from 1 day old to 7 months old. Could you imagine being that experimenter drawing blood from a 1 day old pig? Actually, guinea pig. Oh, that's even sadder because they're really small. Anyway, okay. For science. Okay, scientists incubated an equal number of red blood cells in separate culture dishes. Oh, that'll be the worst job, counting red blood cells in a... Anyway, okay. That contained a 300 nanomolar solution of radioactively labeled glucose. The amount of radioactively labeled glucose present inside the red blood cells in each group was measured over time. Now it says describe a difference between passive transport and active transport. So this is just a fact question, and it's from chapter 6, membrane transport. Remember, passive transport is one that does not require any energy, follows the diffusion gradient, so it includes things like facilitated diffusion and even simple diffusion. Whereas active transport is the one that requires energy, and the main difference, I would say, is that passive transport moves materials from an area of high to low concentration, while active transport moves materials from area of low to high concentration. Now, justify why the scientists used an equal number of red blood cells in each culture dish. Well, because we're using a finite number of glucose, so if you had more blood cells, then more blood cells would have to sort of divvy up that glucose, and you would probably see less radioactivity. And that's sort of not what you want to do. You really want to see the effect of the cell's ability, not the number of cells. So the cell's ability to uptake glucose, you'll make that into a fair game. So you want to have the same number of red blood cells. Glucose transporters are required for the facilitated diffusion of glucose into blood cells or red blood cells. The scientists claim that the expression of the gene encoding for these transporters decreases as guinea pigs age. So this is kind of cool. This is saying that over time, as organisms age, our gene expression can change. And this is actually absolutely true for anyone who is lactose intolerant. Our gene to break down lactose is supposed to sort of phase out as you get older and stop relying on milk. But of course, lactose persistence, a very useful mutation for humans. So most of you guys probably aren't lactose intolerant, but it's the way it's supposed to be. Now it goes, if the scientist's claim is supported by experimental data, predict the effect of increased age on the amount of radioactively labeled glucose present inside the cells of each group. So in this scenario, we would say that the older groups, say 7 months old, would probably have fewer glucose, whereas the younger ones would have more glucose inside the cell. And why is that? Well, because as you age, or as the guinea pigs age, you have less gene expression. Less gene expression means less proteins. Less proteins means less glucose transporters. And that would mean that we have fewer spaces through which glucose can come into the cell. Now, guys, why is it that glucose can't come into the cell without these proteins? Because glucose is a polar sugar. And our cell membranes have nonpolar lipid tails that form the bilayer. So they can't come in at all without this transporter. Now, you don't have to say that here, but I thought it would be something that you guys probably should know. Moving on to the next question. The common wild oat is native to regions of Europe and Asia, but is an invasive species in central California grasslands. In California, the common wild oat has almost completely replaced some species of native bunch grass. Researchers found that aphids, a type of small insect that often carries plant viruses, have a much higher reproductive rate in grasslands that include the common wild oat than in grasslands composed of only native bunch grass species. So this already tells you, by the way, that having these invasive species is bad because you have more aphid proliferation in these non-native species than you would in the native species. And it goes like this. Additionally, the viruses carried by the aphids appear to affect only the native bunch grasses and not the common wild oat. So that's a double whammy. That's terrible. Native bunch grasses infected by the virus have much higher death rates than do native bunch grasses that are not infected. Well, of course, because they're affected by this virus. So this is really bad. Okay, so there's a lot of relationships here, but I'm going to guess that they're going to make us tease out each relationship at a time. So let's go over to A and take a look at what they say. Describe the change in the resilience of an ecosystem when there is a decrease in the number of species. So a number of species here of an ecosystem or of a community is another term for species diversity. And diversity is generally good for a community or an ecosystem because you have more species that can fill various niches, and perhaps a local extinction of one species may not collapse the entire system because you have other species with similar niches that can easily take over. So I always like to think about the number of species as pillars that hold up a tower, or I'm sorry, not a tower, but like a bridge. So if you have a bunch of pillars that hold up a bridge, even if one goes, not the end of the world. But if you have very few species, that's like having one or two pillars to a bridge. And if one goes, then the whole thing just collapses. So that's what you want to get to here. B goes, explain how the addition of common wild oats affect the number of native bunchgrass plants that can be supported by the Californian grassland ecosystem. So I think this question is really asking about the limited resources that we face in a finite world. So for example, even though plants use sunlight, which is kind of an infinite source of energy, I mean, we're not gonna be seeing the extinction of the sun anytime soon. That's not all there is to it, right? There's space, there's water, there's nutrients in the soil. And the idea is that these invasive species typically are pretty good at occupying these new areas and proliferating pretty quickly. So of course, the addition of common wild oats is going to negatively affect the native bunchgrass plants because there's a finite amount of resources available for all of them to simultaneously survive. Going on to C, researchers suggest adding ladybugs, predators of aphids, to the California grasslands. Predict the effect of adding ladybugs in the abundance of native bunchgrass population. So I feel like it would help because, again, this is what we call a biological control. Sometimes this can become a problem of its own, but if we just look at it very, very approximately, then we say, all right, ladybugs are going to eat the aphids. The aphids carry this virus. This virus seems to affect native species of the grass, bunchgrass. So hopefully by alleviating the viral infections, you would initially already get the bunchgrass species to come back a little bit. So no matter how you spin it, this is going to be a positive. And that sort of answers D as well. Okay, ecology, that's fun. Let's move on to number 5. Number 5 says, researchers study mechanisms that enable or prevent speciation. Okay, so now we're moving on to Unit 7, which is on evolution. Specifically, I think we're looking at macroevolution, somewhere around there. Speciation kind of teeters the line between the two. Actually, that's why it's so hard, right? Because we have to scale up microevolution in order to get to macroevolution. But anyway, A says, describe a post-zygotic mechanism that prevents gene flow and thus enables speciation. So we have a lot. Post-zygotic in this scenario refers to the fact that you can create a zygote, as in the fertilization of the egg by the sperm occur between the two species, but something is holding them back from continuing this lineage. And that would be things like hybrid sterility. So for example, we might be looking at horses and donkeys producing mules that are sterile. We might be looking at hybrid breakdown or hybrid inviability. All of those are going to be great answers to the prevention of gene flow. Now, the next part goes, new genes can evolve from non-coding regions of DNA. It is not until certain regulatory elements are present in the DNA that a non-coding region becomes a new functional gene that encodes a protein. So what this means to me, though, is that, well, these non-coding regions were just non-coding by function, but they actually still contained useful data, right? Data that was sufficient in producing a sensible three-dimensional protein. Okay, so what are these regions? So we have promoters, okay, 5' UTRs, and we have a start codon, we have the 3' UTRs, and actually this is 5' UTR following the start codon. Start codon is certainly part of the gene, and then we have the 3' untranslated region too, and you can see this here. And in the example that we see something called AG gene, which I hope they'll tell us what it is. And holy crap, look at that. Okay. Let's read on. All right. Researchers study the evolution of the family of antifreeze glycoprotein. Okay, so that's what AG stands for, antifreeze. So right away I'm thinking, okay, this is going to help you survive in the cold. Encoding genes in Gattidae, a family of marine fish known as cods. I love cods. Okay, when present in the fish, these glycoproteins reduce the freezing temperature of the fish, and these research... Oh, I'm sorry, the video wasn't capturing the text. That's my bad. Let's do that again. Okay, so researchers study the evolution of the family of antifreeze glycoprotein encoding genes in Gattidae, a family of marine fish known as cods. When present in the fish, these glycoproteins reduce the freezing temperature of the fish. The researchers compared genomic sequences in 9 cod species and 1 non-cod species known as B. brosmi. Brosmi. That sounds like words that you guys use in the classroom. Anyway, they recorded the presence or absence of the elements of functional AG genes as well as AG-like sequences that are similar to a functional gene but have undergone mutation and do not contain all the elements required to enable protein production. So that's labeled in Figure 2. So in Figure 2, here we're looking at a phylogenetic tree that actually has timescales on it. So that's kind of cool. And over to the right, it looks like the first column is promoter. We have the AG, that's the functional gene. And then we have the 3' UTR. We have all of these different species with the brosmi fish. That's, of course, our outgroup. And it seems like they live in warmer temperatures. And as we go downwards, we have 1.6 to 6 degrees. These are cooler temperatures. And as the key shows, and I think you guys probably have figured this out by now, the pluses would mean that the promoter is there for these guys, it's absent for these guys up here, and so on. So you can see already that the box that contains all the fish that survive in these colder waters, they have all the elements that they need. But anyway, let's go ahead and take a look at what they say. Based on the data in Figure 2, explain how changes to the genome enabled cots to survive and reproduce after a period of freezing temperatures between 10 and 15 million years ago. So here we have a bit of an ice age. The changes to the genome that enables the production of these antifreeze glycoproteins, how would that be helpful? Well, of course it would be helpful because they're now able to survive in the cold temperatures. They spend less energy trying to thermoregulate. They can allocate that energy for reproductive endeavors, and that would mean that they would leave more babies behind. The ones that have that mutation would leave more babies behind, which means that it proliferates very quickly through the daughter lineages, and that's clearly what we see in these 4 species. Using the template in the space provided, place an X on the phylogenetic tree to represent the origin of the functional AG gene. The origin of the functional AG gene, as this little box shows, AG repeat sequences, and of course, all of the promoters that are functional would be right here in this line. I'm not sure if you guys can see that. Okay, maybe I can just use video editing to put that X there, but it's going to be right there because all the descendant species have everything. Now, just for a quick reminder, this little M. merlangus over here with the minus, that probably is a separate mutation that occurred later in the lineage, losing the 3' UTR. It doesn't mean that these two evolved and that one didn't. It's much more likely, and what we call parsimonious, much more likely, parsimonious, to say that all of these kicked in at the same time and merlangus lost it right around its own lineage later. Okay, so far, not bad. Based on Figure 2, explain how genetic differences among the species of Gattidae family determine the habitat in which they can survive. Wow. So thankfully, we don't have to kind of disseminate this entire thing, but it's a very simple question. So the genetic differences, so the ones without the antifreeze proteins, well, they live in temperatures that are warmer. Ones with the antifreeze proteins live in much cooler climates. And this could be, you can extrapolate this to latitudinal differences. Say these are Alaskan cods, Russian cods versus cods that are found in the warmer Pacific. I'm not sure. I'm just a big connoisseur of eating the cods, but I don't know where they live. I was a plant guy. Moving on to the last question. Wow, this looks like a fun graph. Scientists can quantify the rate of translation as ribosomes move along an mRNA from one codon to the next. So here we're looking at gene expression, chapter 17. Using a procedure called ribosome profiling, the scientists measure how long a ribosome remains stationary at each codon of each mRNA. They determine that the average translation rate across all codon is 5.2 amino acids per second. That's pretty fast. But that the average translation rate for specific codons in different mRNA sequences can vary widely. These variations in translation rates are thought to facilitate correct folding of the protein being produced. So you guys know that as the polypeptide is formed, they begin to fold right away. So maybe giving some breaks to make sure that the folding occurs properly might be built into our evolution. So molecular evolution, that's cool. The rate at which these 3 different codons were translated was measured in 100 different mRNA molecules. The scientists determined the distribution of rate, number of times each rate was recorded. That's important for each of the 3 codons, GAC, AUU, and UGG. Now notice how you don't have to know what these do. I think that's phenylalanine, but it doesn't really matter. Like, I don't know what GAC is. It's okay. Now, the idea is the more important part is that this is a frequency distribution, meaning that every time that they recorded the amount of time it took to place that GAC down, or the right, let's say, amino acid down, they recorded how long it took, and the frequency of those times are recorded here. And there are a few things that we learn from these frequency distribution charts. For one, for example, when it comes to GAC, the most recorded time was 150 millisecond per codon. And then we have a distribution like this that sometimes it took up to 650, like 1 or 2 times. And then here in AUU, majority of, the time it took was between 100 and 150 seconds, and nothing went over 450. So the spread of the data is tighter here. Now, the spread of the data is way wider here, as in the variance, right? The amount of time it took to place down UGG, it It wasn't like, oh, majority of the time it was 200. It was like, well, sometimes it was 50, sometimes it was 800, sometimes it was 950. More often than not, it was between 200 and 500. But you can see that the spread of the data is easily visible from looking at frequency distributions. Okay, so let's look at the questions. Using the data in figure 1, graph A, identify the rate that was recorded the greatest number of times for the GAC codon. Yeah. So they're giving you a break here, right? That's pretty easy. 150 milliseconds. Okay, that's it. Using the data in figure 1, graphs B and C, describe the variation in translation rate of the AUU codon compared to the UGG. Variation, there we go. Variation is how much spread there was in each of the recordings that you took. So for instance, we would say that the variation in the translation rate of AUU was much smaller than the variation of UGG because, well, there's a couple of things that we can look at. We can look at the range, 50 to 450, 50 to 950. We could also look at the spread of the data because we have that information. So you can describe that and we'll be good to go. Scientists hypothesize that tRNA molecules that bind to UGG codons are available in lower abundance than tRNA that bind to AUU codons. Support that hypothesis using figure 1. Now, UGG being in lower abundance, UGG being lower abundance makes sense. Because look at this. Sometimes we needed to wait a long time until that codon UGG could be matched to the tRNA that contains UGG. Now, once again, if you haven't done molecular biology quite yet, you might be really confused as to what I'm talking about. But once again, if you are watching this video in April or May, then you would know what I'm talking about. tRNAs come into the ribosomes through what we call the amino acyl site, but you know that majority of the time the wrong ones are coming in. And it's only when the codon and the anticodon match that you have the proper placement of the amino acid. So the fact that we have so long of a time that passes before the right match is made could be indicative of the fact that you have a very low concentration in the cytoplasm of the tRNA that contains, well, in this case, the anticodon to UGG. So, well, that makes sense to me. That's great. That's a great question, actually. Fantastic job, College Board. Now, the last question says, amino acids can be encoded by multiple codons. In many organisms, certain codons for the same amino acids occur more frequently in an mRNA than other codons. Based on the data provided, explain why the use of one codon over another for the same amino acid might result in increased level of protein production from a particular mRNA. Okay, so we are talking about the redundancy effect here. We're talking about the redundancy. So if we're talking about the idea that certain codons are going to take a less amount of time to place down the amino acid, then it would actually mean that even with those redundancy, you would still likely see the codon that is faster over another redundant codon because that would allow you to produce that protein faster. And that's really all there is to it. So even though there are the same codons that are redundant, we see some of them taking precedence over the other due to the efficiency factor. And that's it. I think this is kind of the hardest question from this set here. All right. Anyways, so I'm not sure how long this took. I have a feeling that it's a little bit more than half an hour, but I was pretty close to it. And hopefully I was as comprehensive as I could be in explaining each of the questions. And well, not the concepts necessarily, because there's, it will never end if we went into all the nitty-gritty concepts. But if you've done the concepts, hopefully this video was useful. If not, then please pause and reference those chapters or units that I correlate with each of the questions, and that will help you in contextualizing why the AP is asking those questions. So this was our long-form video, and if you like this kind of content, let us know. Leave a comment. Say, hey, more of this. Less of you fast-talking on those short-form videos. I enjoy making both, actually. They're both kind of fun, but let us know what you like. And if you haven't done so, click like and subscribe so that you can stay on top of more videos like this. This has been Mikey with Avo Prep Academy, and we'll see you in the next video. Take care.